Hello there. It is I, Gregor Johann Mendel. I was born in my beloved Austria, which is now the Czech Republic. In 1822, I excelled at school, of course, and a priest actually advised me to study even more, so I went to the University of Vienna. Then, when I graduated, I became an Augustinian monk, against my father's will. Actually, the name Gregor isn't even mine. It was given to me when I entered the monastery. There, I began my studies with genetics and heredity, and my most renowned experiment with my beloved bees.
Mendel worked with mice, honeybees, pea plants, and flowers.
Mendel's experiment with bees consisted of him first crossbreeding a different variety of purebred pea plants. He then collected the seeds and grew them and analyzed the offsprings. The results were not a blend. Next, he crossed the offsprings with each other, which is called self-fertilization. With this, the resulting progeny had two different traits, tall and short.
The results of his experiments can be seen in this table: purple and white, tall and short, yellow and green, round and wrinkled, green and yellow on the side, full and flat, axil and terminal. The first ones are the dominant traits, and the second ones are the recessive. His conclusions were that organisms had factors that determine its features, now known as genes. We also discovered that organisms possess two versions of each factor, alleles. Also, each gamete contains only one version of each factor. Parents contribute to the inheritance of the offsprings, as the unions of sperm and an egg make a randomization of their genes. Finally, for each factor, there is a dominance that will always be expressed if present a recessive one.
From these conclusions, rules were made. Mendel established then three laws. First, the law of segregation. When gametes form, alleles are separated so that each gamete carries only one allele for each gene. Second is the law of independent assortment. The segregation of alleles for one gene occurs independently to that of any other gene. Lastly, the principle of dominance. Recessive alleles will be masked by dominant alleles.
Were friends. They did not believe I was a true scientist. Even I did not believe my experiments were of great importance to biology. I thought I was only working with hybridization. I never imagined I would be known as the father of genetics. I did not promote my work at all. Oh, well, yeah, a little bit. And unfortunately, most of it was burned by the church after my death. It was only after hundreds of years that my work surfaced. And only until then did my findings impact scientific knowledge about genetics and heredity.
An offspring can have different traits from its parents because it is often a combination of genes that give an organism a particular trait. Because there are so many possibilities when two parents combine, we can use a tool called a Punnett square. A Punnett square is a graphical way of determining all of the possible genetic outcomes when a cross is performed. It's a probability box that shows the odds of each possible trait happening. When genes are demonstrated in a trait, one of them is from the mother and the other from the father. Genes can be dominant or recessive. When they combine, there are three possible conditions: capital B homozygous dominant, capital B heterozygous, and lower B homozygous recessive.
In this first Punnett square, we are combining the genes from two heterozygous genes. The combinations will be 1 homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive. In this second Punnett square, we are combining the genes from 1 heterozygous and 1 homozygous recessive genes. The combinations will be 2 dominant heterozygous and 2 dominant recessive.